So in this video we'll discuss Yersinia infections. So the tribe Yersinia comprises of genus Yersinia, which has three well-established human pathogens. So Yersinia pestis, so it is the agent of plague, a fulminant systemic zoonosis transmitted from rodents by an arthropod vector, the rat flea. Okay, the rat is a rodent. It is transmitted from the rat by a rat flea. Yersiniosis is a zoonotic infection caused by the Enteropathogenic Yersinia species, the enteropathogenic. So Yersinia enterocolitica or Yersinia pseudotuberculosis. Pigs and other wild domestic animals are the usual hosts, and human infection is by eating that contaminated raw pork. Okay, most common in childhood and in colder climates. So obviously, where are the areas you expect to find it? So colder areas, right? So Northern Europe and America. Why enterocolitica is found worldwide, but why pseudotuberculosis are generally rare and have been reported from Finland, cold countries. Okay. So based on serogrouping, so why enterocolitica is further characterized biochemically. It has 6 biotypes, and then based on the O antigen, it has 60 serotypes. And worldwide, most clinical infections are associated with serogroups O3 and O9. Next, why pseudotuberculosis? It is further differentiated into 6 subtypes, 1 to 6 based on both somatic O and flagellar H antigens. So clinical manifestations: yersiniosis, SSS, self-limited gastroenteritis, septicemia, and superantigens. So soup, and self-limited gastroenteritis. Remember this, self-limited gastroenteritis doesn't need treatment, but septicemia needs treatment. We'll discuss that later. So laboratory diagnosis: for isolation from blood, use blood culture bottles. For isolation from lymph node aspirate, culture is done on conventional media, so blood agar, nutrient agar, and McConkey agar. McConkey agar is also selective. So for isolation from feces, food, or soil, selective media is used, such as deoxycholate citrate agar, DC agar, and McConkey agar. Lab diagnosis: plates should be incubated both at 25 degrees and 37 degrees Celsius to differentiate it from most of the other pathogens, which grow only at 37 degrees Celsius. So cold enrichment: Yersinia likes cold countries, right? So cold enrichment can also be done by incubating in phosphate buffered saline at 4 degrees Celsius for 3 weeks. Okay. Next, why enterocolitica and why pseudotuberculosis show the following properties by which they can be differentiated from why pestis. So differential motility. So both these enteropathogenic Yersinia are motile at 22 degrees, colder temperature, but not at 37 degrees. Cold enrichment: so growth improves on refrigeration at cold temperatures, so 4 degrees Celsius for 3 weeks. So cold enrichment. Automated identification systems: so MALDI-TOF, mild. Okay, so MALDI-TOF is used for accurate species identification. So lab diagnosis: antibodies can be detected by ELISA using serotype specific coantigen types. And why pseudotuberculosis infection antibodies appear early, okay, during acute phase of illness, whereas enterocolitica specific agglutinating antibodies appear in the convalescent sera. And finally, the treatment of yersiniosis: most cases of diarrhea are self-limiting. Treatment is required only for systemic infections, such as in case of septicemia. So what do you use? Fluoroquinolones like ciprofloxacin or third generation cephalosporin such as cefotaxime. Okay, why enterocolitica strains nearly always produce beta-lactamases, but not why pseudotuberculosis strains.